So number 4 then, biogeography, which just means the distribution of species across the globe. This gentleman is Alfred Russel Wallace, and like Darwin, he was a naturalist and was thinking about ideas of natural selection at the same time as Darwin. In fact, he started a bit later, but came to his conclusions a bit quicker. And in 1858, Alfred Russel Wallace was going to publish a paper on natural selection. Now, very fortunately for Darwin, they had a mutual friend called Charles Lyell. And Lyell talked to both Darwin and to Wallace and made the fact that they were both thinking in parallel known to both of them. Darwin writes, "I never saw a more striking coincidence. So all my originality, whatever it might amount to, will be smashed." Now, Wallace, fortunately, although he had the upper hand, was a gentleman, and they agreed to publish their first paper together in 1858, the year before Darwin finally got everything together for his book on the origin of species. So natural selection really should be known as the Darwin-Wallace theory, and there's a word there for Alfred Russel Wallace. Bill Bailey is an expert on Alfred Russel Wallace, gives lectures on him actually. Anyway, both Wallace and Darwin came at the idea of natural selection from a slightly different direction, although their ideas overlap very broadly. Wallace was more interested in the natural distributions of species and the fact that they were adapted to different environments in different geographical regions. Darwin also thought about this in detail, but he emphasised a little bit more competition between individuals for resources, but both had ideas that overlapped. The basic bit of evidence from biogeography for evolution is that we don't find species living in all the places where they would be perfectly well adapted to thrive. And the reason we don't find them everywhere is that they originate or they evolve somewhere in particular. They have a centre of endemism, and they may radiate out from that centre of endemism until there's a barrier like an ocean or a change in some physical condition that means that they're no longer terribly well suited. But there are lots of species that we can transplant from one part of the globe to the other, and when we do, they thrive and they do extremely well. In fact, they may even do better than the species that are endemic there, or the native wildlife. Some of these are called invasionary species, and a good example of that might be rhododendrons or grey squirrels. So grey squirrels are not endemic to the British Isles, they've come in from elsewhere, and they've outcompeted the red squirrels. So they do very well here, thank you very much. Here's another example. The thing on the right is a cactus, and this is from the Americas, and the thing on the left is a euphorbiid. Aside from being an excellent example of convergent evolution, these are two rather different groups of plants. They are found in different regions of the world. If you transplant them to one another's regions, they do extremely well, they do perfectly well. The only reason that they're not found there naturally is because they originated elsewhere and they were unable to spread to the other region. This is a view of the Mediterranean, and the Mediterranean opened up and became a sea about 5 million years ago or thereabouts. And so the animals living in the Mediterranean, of course, have come in through this strait here from the Atlantic. And although this is a shallow, warm sea, and this is a cold, rather deeper sea, the fauna and flora here much more closely resemble that in the Atlantic. Rather than, for example, down in the Red Sea here, which is also shallow and warm like the Mediterranean, but the fauna is different. Now, the animals living here would do extremely well in the Mediterranean, but until the construction of the Suez Canal, they'd never been able to make it in. So things are where they are because of evolutionary history, and things actually evolve in a particular place. So the fifth bit of evidence for evolution is direct observation. We can actually observe selection taking place. If you think about it, we've selected our pets, dogs and cats. Dogs in particular are very plastic in terms of their form. We've selected them to have traits which we find agreeable. The same with our livestock. We've selected them to be placid and to produce lots of milk or food for us. Cast your mind back, if you would, to 1185, and there are two large fleets facing off across the inland sea in Japan, the Bay of Danura. One fleet belongs to the ruling royal family, the Heike clan, and the other fleet is that of the Minamotos, a warring tribe that wants to take control of the throne. The emperor of the Heike is the 6-year-old Emperor Antoku. There's a huge battle, there's a huge loss of life on both sides, but eventually the Minamoto clan get the upper hand. Rather than surrender, the 6-year-old emperor and his grandmother plunge into the sea and they're drowned. Now, this is a piece of history, but it's also gone down into legend. And the legend is that the souls of the Heike clan entered the bodies of the local wildlife, and particularly a species of crab. Now then, imagine that you are a fisherman and you catch a crab, and purely by chance you happen to look at it before sticking it in the sack to have for your tea or to sell, and you notice that the carapace of the crab looks very, very slightly like a human face, only a little bit. Maybe you throw this one back rather than eating it, because maybe you're a little bit superstitious or just a bit curious. Over time, this is artificial selection. The ones that get thrown back that look a little bit like a human face get to have more baby crabs. And as long as the selection is sustained, then the face potentially will get more and more like a human, until you end up with something like this. This is the carapace of the Heike crab, so called, and it looks like a scowling human face. This is artificial selection at work, human-based selection. And of course, the same principle applies with natural selection, but we can see the mechanism playing out. One final word then, and that's on one of the detractors from the idea of evolution, and it's something that's come up in the last 30 years or so called irreducible complexity. It's the idea that there are some structures in biology that are so complex that they couldn't possibly have evolved. The argument goes, look at something like the human eye. It's fabulously complex, fabulously useful, of course, to be able to see, but it's made up of lots of different parts. It's got a lens, it's got a cornea, it's got gelatinous material inside the eyeball, it's got a retina that's made up of layers of cells, all immensely complex, wired up to the optic nerve and to the brain. Now, if any part of that structure, for example, the lens, if that clouds over, you can no longer see. If you have a problem with your optic nerve, that might also mean that you can't see. And the argument goes that you need the whole structure in one go. The whole thing has to evolve as if by a puff of magic in order for that structure to have any function or to be of any use to the organism, to be of a selective advantage. And that sort of macro mutational evolutionary leap to evolve all of the parts at once is, quite rightly, or would be, fantastically unlikely, and therefore very unlikely to happen. And the idea of irreducible complexity, therefore, is that there are these structures that are too complex to evolve. We can't posit intermediate stages that would be functional. Now, of course, that's true of your eye. Remove any component, and yes, you can't see. But there are animals, if we look down the tree of life, that have eyes of a variety of kinds. So you can have an eye like ours that has a lens and a retina, but you can also have, for example, a pinhole camera eye, an eye that doesn't have a lens, that forms an image. It's not perhaps as good as one that would be if you had a lens, but nonetheless, it's very useful for the animal that has it. Or you could have an eye that's just a simple cup of light receptive cells. This might allow you to see what direction light's coming from, or it might allow you to see where a shadow's being cast. That could be a predator, and you could get out the way. So this could save your life. It's therefore of huge adaptive advantage. And there are even precursors, if you like, of eyes that are just light sensitive patches, and again you can tell, or the animal can tell, if a shadow's been cast over it, and it can respond accordingly. So pardon me, these for example are some of the eyes around the mantle of a bivalve mollusk or a clam, and these are very simple eyes. They allow the animal to see if something's coming to try to get it, and the animal can then close its shell. This is a nautilus, so a mollusk. It has a cephalopod mollusk. It has an excellent eye. It's a pinhole camera eye. It forms an image, but there's no lens in here. And the eyeball, if you like, is filled with seawater. But it works perfectly well for it. This is a flatworm that has a very, very simple cup-like eye in here. So two of them. And these cups allow it to tell which direction, pretty much left or right, the light is coming from, and it can respond accordingly. And this is a single-celled protist called a euglenid, so just one cell here. The eye isn't the red spot, but the eye is just alongside of the red spot, and the red spot casts a shadow on the eye, or the light-sensitive organelle, and it can therefore respond to the direction of light and the intensity of light. So these are not eyes as such, I should say, in this euglenid, but they are light-sensitive structures that are useful to the organism in question. So we can easily posit a series of transitional types of eyes up to the most complex. There are other types of eye, of course. This is a complex compound eye from a crustacean, but there are other there are other arthropods that have much simpler compound eyes. This is a millipede. You can't count the facets here, but you can here. And you can go on building more and more complex eyes of this type. One more example. You probably know that in your middle ear there are three bones, the hammer, anvil and stirrup, and they amplify sounds from your eardrums onto the cochlea, and it gives it a great deal more resonance in the signal. Now we know, again, from transitional fossils, but also from embryology, that those bones originate as part of the jaw, and that in developmental terms and also in evolutionary terms, those bones have migrated from the jaw and into the middle ear. Again, we have this lovely transitional series of fossils for mammals to show this. So here's how we've put together a complex levered structure from something that was once in the jaw. One more example of supposed irreducible complexity, and I'll stop there. Bacteria have a flagellum that they're able to rotate. It's pretty much the only wheel that I'm aware of within organisms. And this flagellum is made up of a number of components. It's a bit like a motor at the base, with this thing sticking in here and an angle, and then the flagellum that sticks out here. And this whole thing in here rotates, goes round and round and round, exactly like an electric motor. It's made up of a number of components, and if you take, again, any of these components away, the whole thing breaks. And the irreducible complexity argument would be that you can't evolve the whole thing in one go, and the thing is useless without all of the parts. Catch-22. So therefore, there must be some other form of intervention other than evolution. Actually, what it turns out is that many bacteria have components of this system, but they're using it in order to attack eukaryotic cells. It's a so-called injectosome. So this is the injectosome from Yersinia pestis, or the plague bacillus, and it's got many of the components that the motor has, or the flagellal motor has, but they're doing a different function. They're doing something else. And so you can see that it's possible to evolve a more complex structure with a new function from something that's doing another job, in this case, allowing the animal to parasitize or feed on eukaryotic cells, or allowing the bacterium to feed on eukaryotic cells, I should say. So that's it, I think, for this particular session, and thank you very much.